So today I will be talking rather in general sense and with few details to give you my view how the metabolomic is impacting biomedical research and what are the present challenges. I would like to introduce as well a new German Society for Metabolomic Research, which was founded last year. Let's start with cohorts. Of course, cohorts in human studies are the most important tool. I know that many of us are using cell culture and animal models, but when the health is concerned and when the disease is concerned in humans, there is nothing like decent cohort research. So there are different types of cohorts, and they might be considered as proxies to that what we are looking after, the phenotype description or phenotype explanation. Definitely other cohorts used a cross-sectional approach, like Farmigan study, located in one place, located around one job, nurses, for example. With the time, it was clear that prospective character, that a collection of samples of the same individuals is of great importance to the explanation of phenotypes, because some phenotypes develop with time. There are as well disease-focused cohorts. They are usually smaller because it's not possible to control the patient description if you're using too large cohorts. Of course, this is not only the size, this is the depth of the phenotype description, which is giving the quality of a cohort. We are facing right now different tendencies in the world. For example, prospective longitudinal cohorts and cross-sectional cohorts are fusing. This is a challenge for logistics, and this is a challenge as well for the compliance. I don't think it is easy to recall the individuals over years, but it is a way we would like to see the phenotypes being developed. Intervention studies is something where the mechanistic studies could be done, for example, challenge with a diet or challenge with a drug or challenge with a sport. And they are providing more focused information about the mechanism behind the phenotype which is being analyzed. So all of that has been used for omics and especially for metabolomics, and there are some requirements which metabolomics has, and I will address it in the next few slides. We all know that the word omics is originating from Greek rho, omos. Actually, it started with genomics, with gene research. Nowadays, we know as well that the knowledge how to deal with large data, how to design cohorts, is different for different omics. And this is because of one very important factor. This is the number of levels, freedom levels, okay? So in the omics, called genomics, for example, we have actually 4 levels of freedom. This is ATCG. This is modified by indels, CNPs, copy number variations. This is modified by epigenetic events. But actually, starting with these 4 levels of freedom, you can get a lot of information, which is very unique and very specific. Now, let us think about metabolomics. It holds as well for proteomics and for lipidomics. We have the possibility that a given metabolite is not present at the moment when we are measuring the sample, that the concentration may be different at different days in the same individual, that the concentration in different tissues will be different. Of course, I'm referring rather to animal model than to human, but this is the case. So these are all levels of freedom for metabolite. To make it worse, the same metabolite could be present in different pathways. So even giving the information that you know that the metabolite is there and you know the kinetics, it's not very clear without a decent, stable isotope fluxomics to find out where is it coming from and what is going on. Now, there are some challenges in the metabolomics which are interconverted and reciprocal. So, for example, it would be beautiful to have sample coverage with 4,000 metabolites, but at present it would take roughly 1 day if you would stick to one method, for example, very long column, LCMS, and so on. On the other hand, if you would go for only 4 metabolites or 5 metabolites, it could be possible to measure them in a few seconds, because you could optimize them for much shorter analytical procedures. Of course, in the case the high coverage is preferred, the costs per sample are very high, because you cannot transfer process the many samples a day, and this is why the costs are very high. There are some approaches in which several thousands of samples with one metabolite are measured, and the costs are really cents a day. On the other hand, standardization with few samples is relatively easy. The level of errors could happen during the procedure is something could put down very, very fast. In the moment, the many metabolites and many samples are coming into the play, even as there are throughputs which are working well, the comprehensive description makes the story difficult. So there are reciprocal and interconnected trends. At present, we would like to have a sample with measurement which has a lot of metabolites, which is very affordable, and which is standardized. And this is bringing me to a summary of requirements which I put together on this slide, which are applicable to different cohorts. That's why I cited cohorts in the very beginning. So we are looking for methods, or a method with a comprehensive metabolic coverage. If we were running assays for a large cohort, it should be kept constantly at the high coverage for all samples. It should be reproducible and robust, in the sense that even if we have the option to run thousands of samples, it will be the same quality. Sustainable is something which actually epidemiologists require. So they would like to have the same method over years, because not every cohort could be measured at the same time. One study stimulates the other one, and the studies without a comparison of direction of changes are not really dependable. The requirements for the metabolomics tell us that the methods should be accessible and affordable. Accessible in the sense that if there is a method running in-house and you cannot send your samples in, this is something exotic and probably will be not sustainable. Affordable is something which is always bothering me, that we never have enough money to spend them on a decent research, which would be covering all parameters which are requested by the study design and power calculation, because we don't have enough funds. Throughput matching demand is something which is obvious if we calculate that let's say cohort of 5,000 samples would need 1 year to be measured. Then you think, okay, seasonal variations, people going for vacations, pandemic, whatsoever. So this is something which is always projecting into the looking for a provider or collaboration partner or laboratory within the institute. Standardization is something which is becoming, and I love the presentation today from Robert Gorka. This is something which is absolutely the beginning of the decent study. Without that, it is difficult to depend on the best statistic. Scalability is projected from standardization, because we could make copies on standardized assays and use them to create more throughput. So this is my vision about the requirements for metabolomics in cohort studies. Replication is that what we are looking for: large, medium, and small cohorts. They should use these all parameters to connect the different cohorts worldwide, different animal models worldwide, to allow data replication. Now, let me give you a few examples. I think the metabolomics is providing more information that genomic does. I'm not saying that genomic is wrong. I'm saying it is not explaining the phenotypes we're analyzing. So in this example, I have taken it from the paper of Ciroli, 2018. The hypothesis was that we are looking for the factors which are explaining actually body mass index in a given population where there are people of different phenotypes, from lean to obese, and they are as well stratified for the insulin resistance, genetic risk, and cardiovascular events. So in the population of interest, there were 4 groups found by this stratification, and it became very clear that the BMI could be very fast and very dependently explained by the metabolomics. It was an explanation of 50% of these phenotypes, whereas genetic explanation of this variability or this stratification, excuse me, was less than 3%. You should know that one of the authors of this paper was Greg Venter, so the guy who actually promoted human genome sequencing. And I think this is very good to see this prominent scientist saying, okay, these are the limits. Let's go farther with them. My next example, I would like to cite the data from the Dunbar. I was happy to be a member of this team, in which another question was asked. If we have a general population with different age and different phenotypes, how can we explain variability of the serum metabolome? This was an experiment in which metabolites have been compared with different factors known for this cohort. They were diet, clinical data like blood pressure, for example, microbiome diversity, not the amount, but diversity, genetic diversity, genetics of humans, circadian rhythm, and lifestyle. So here you have the explanation of the human serum metabolic variability, and different aspects given to the left are plotted here. You can imagine that the combined model, full model, including all parameters, explain the variability the best. But I was quite surprised to see that the most contributing parameter was diet and clinical data and microbiome. Probably microbiome and diet are in this case are confounded. Nevertheless, the same in this case, genetics was not contributing much. In the specific models, different classes of metabolites were contributing to explanation, but the most important to me was to see that among the top predictors, there are lipids. And indeed, the lipids, the new classes which we are able to measure right now are expanding our window how to see it. And I'm very happy to see that specific classes like ceramide and only single molecular lipids in these classes are contributing to these explanations. Very important tool in analysis of animal models and human cohorts is the knowledge how good is the method. And we use the ring trials to assess these methods. So there are many which have been published, example, the Biocrates P180 kits, but there are others which are coming on the market. For example, Martin Gera has finished a lipidizer ring trial in which several laboratories were comparing the performance of a lipidizer assay on the same sample. From his work, there is another product which is coming on the market. This is called Lipidizer 2.0. And this is simply open tool, the open source metabolomic measurement, which contains MRMs, procedures, how to prepare the samples, how to put together the standards. And this is all described in the way which would pass the ring trial. There are different approaches how to do the comparisons in metabolomics. For example, Markus Wenck has performed and is writing it together the assays of specific classes, in this case, ceramides and bile acids. I am right now putting together the data, finishing the manuscript in which we have compared the performance of MXP 500 assay, and it has been done across the globe, from Japan, Singapore, through Germany, Canada, United States. So we'll see what is reproducibility, what is the interlaboratory precision, and which metabolites we should take with care into the interpretations, and which are quite dependable across all possible conditions. So I think ring trials are very important in providing the information which assays are good for you. The standardization is supporting very interesting effect, which I think is worth discussing here. At present, different institutions, different research groups are usually testing one cohort or one animal model with all different methods. Nothing wrong with that. Usually out of such a study, a lot of data is showing, and this is providing mechanistic information. This is how the people build up a career and the knowledge is created. However, as I said at the very beginning, the sizes of the cohorts and the complexity of the cohorts, like longitudinal sample collection, is making the cohorts much, much bigger. So not 5,000, not 10,000. We are talking about 50,000, 100,000, even 500,000 and so on. So it is not possible that a single laboratory would be able to handle such a cohort. And there is no industry worldwide which would be willing to perform this risk to make a factory for metabolomic phenotyping. I think the possibility is to perform upscaling and perform simply the parallelization of the assays. That would solve the demand of characterizing the cohort within a reasonable time, not 10 years, maybe a year for 100,000 people. So this is the concept which is very appealing. What would be the advantage of such a standardized assay is, for example, as well, the fact that the results from other cohorts would be used for the replication, simple replication. At the end, I would like to address the point that longitudinal challenge is getting more and more present. Different biobanks are simply collecting the samples. This would allow us to address the effects like inborn errors of metabolism. We know that over 10,000 monogenetic diseases is present already, but they have genetic penetrance, which is showing the phenotype only specific time windows. We don't know why is it, but it should be accepted that we have to address it. Untouched is epigenome metabolome analysis in large scale. This is still in long range because we don't have tools for that. So at the end, as promised, I would like to introduce a German Society for Metabolomic Research. We are incorporated. We have board of directors. The colleagues are coming from different disciplines in the German research area. We have a website, and on the website you can get updated what's going on in the society. We have as our aim focused on the integrative communication for metabolomic research, so we're mediating contacts. We have already working groups on different aspects like standardization, NMR, microbiome. We prepare our own meetings and will take part as organizer of satellite meetings, and all members are welcome. Students, researchers, companies, welcome. This would be my message for today.